All right, welcome folks. We are here for Public Labs live build of the community microscope. So this version that we're going to be building is a dissecting microscope. Vanya's going to show us how to do this. We'll do some introductions of who's on the call right now. So we are recording this event and we'll post it to the Public Lab website later, so you'll be able to see it again. And this event is part of us continuing on with a focus on water quality, specifically wastewater and stormwater. Earlier this week, a couple of days ago, we had a call about some DIY methods and open source methods for monitoring wastewater and stormwater pollution. And we were joined by some community scientists who are doing local projects on this topic as well. Right now, we're going to focus more closely on one of those tools that we talked about, and it's the community microscope. So we're going to do some introductions first, and since we're a small crew, we can all spend some time doing our intro. So you can share your name and where you're calling from, and something that you're interested in seeing today or learning about today. So I'll start, and then I'll popcorn it over to someone, and then we can just popcorn around. So my name is Jeanette Lim. I am the research coordinator at Public Lab, and I am super excited to be joined by several fellows on the call today. I'm going to hand it over to Vanya.
Hello. Hi, I'm Vanya. I'm the Air Quality Research Curation Fellow with Public Lab, and I'm excited to build the microscope and excited to see what ideas you all have to put under the microscope. I'm calling from San Francisco, California. I will popcorn it over to Alana.
Hi, y'all. My name's Alana. I use she, her pronouns, and I'm calling in from New Orleans, Louisiana. The microscope is one of my favorite kits that Public Lab has, but I always struggle to rebuild it every time I try. So I'm stoked to have the group think on this one. I'll popcorn to Laurel.
Hi, everyone. My name is Laurel Meir. I am the Land Research Curation Fellow for Public Lab, and I am calling from a suburb of New Orleans, actually, across the lake on the North Shore. And I don't have any experience with the Public Lab, DIY microscopes. I'm really excited just to see it put together and look at some fun and wacky stuff underneath it. And I will popcorn it over to Amir.
Hi, so I'm Amir. I'm originally from Israel. Right now, I joined my wife in New York City. So it's really nice here, and it's daytime, not night. My background is in physics, and I've been remotely involved with the public laboratory ever since the microscope, the spectroscope Kickstarter a few years ago, and have been following that for some time now. And I'm basically, I enjoy anything that has to do with optics, imaging, and if it's some public science behind it, then it's even better. And I'll pass the ball to Alejo. You're muted.
Hello. Hello, people. Well, my name is Alejo. I'm the Water Research Curation Fellow. I was, I'm sorry. I was writing in the chat to not scare it to Sophia, but I think that Sophia was intimidated and she goes away. I'm sorry, but well, I'm very excited to be here with all the staff and all the people interested in wastewater as Amir. And I'm very happy to be part of this community, and I want to learn a lot of things and to show some things that I was made in this last month.
Thanks everyone for the introductions. So Vanya will be leading the build portion of this event, and then Alejo will show us some applications specifically to this topic of wastewater and stormwater monitoring. So I will hand it back over to Vanya.
Okay, cool. So I'm going to switch over to my overhead cam. Hopefully folks can see all show the materials somewhat in the frame. So I can just give a quick intro of the materials that you'll be needing, and that you'll get all of these materials in the kit. So with the exception of maybe this piece of paper and some tape. So let's get started. So what we have here, you'll see a webcam. We'll convert this to kind of the thing that you'll use to use the microscope. And then we have a couple washers, 6 to be exact, and a couple wing nuts. So we need 9 wing nuts, so 3 more than the washers that we have. And we also have 3 long bolts and then 2 stages. So they might be different materials depending on what version of the microscope you end up getting, but essentially these are just plastic. These are kind of hard to see on the camera. So you'll have 2 rubber bands, and you'll see what they are for, and then 1 light. So so it comes with this like long gooseneck light, but anything with like 1 singular LED works. So I've used my phone flashlight, and that's worked. You can be creative with your light. And then for this dissecting microscope, we will need just a white piece of paper. You can use a Post-it note and just a little bit of tape. Yeah, we can get started. So the first thing to do is to build the stage, which will hold the microscope, and that's actually the bulk of the work. So we can get started. Oh, and one thing is that you will need, and you will also get a small, super cute screwdriver that comes with the kit. So all you need to do, it took me a while to figure it out, but you have to like unscrew it, and then it will have 2 sides. Unscrew it properly. Yeah, and so you have 2 sides, depending on which side you need. Yeah, for the stage, you start with these 2, and you'll see they're pretty similar, but the main difference is that one of them has a circle and the other one has 2 like rectangles. So the 2 rectangle one goes on the bottom, and the circle one goes on the top, because that's where eventually your microscope will be like this. You'll put it on top. So start with this one, and you just put your wing nuts through it. And let me know anytime if the video if I'm like out of the range of the camera.
You're looking good, Vanya.
Okay, great. Look, cool. So now that we've put all these through the holes on the side, so there's 3 holes. So the 3 holes match the 3 holes on the top one, because you pretty much just stack on the top of each other. And then the next thing to do is to screw them in with washer. Make sure to put the washer in first, and then do the wing nuts. The first time I did it, I forgot to put the washer in, so I had to like take it all out. So don't do that. And can also make sure that it's right side up and not upside down. The right side up has some numbers. I don't know what the numbers mean, but it just means that you should put it right side up with the numbers facing up top. And then all you do is screw in the wing nuts into the long bolts. So while I do that, so Jeanette mentioned that Jeanette mentioned that this is a dissecting scope. So you may already be aware, but for folks who are not aware, there are kind of 2 general types of microscopes. There's the compound microscope, compound light microscope, and the dissecting scope. So the compound light is where the light source and the camera are on different sides. So it's really good for seeing really thin specimens. And essentially the light has to pass through the specimen to get to the camera. And so it's good for kind of thin specimens and cross sections. And this dissecting one is much better for 3D specimens, where it's a little bit larger and it's too thick for the light to pass through. So you do end up creating the light microscope configuration, just be aware that don't put things that are too thick, otherwise you won't be able to see anything. So for this configuration, you can put things that are not see-through. So once we have these nuts secure, just tighten them, and then this, yeah, it seems pretty tight. It's not gonna fall off. So next thing is you will put this other stage on top. Actually, before we do it, it would be smart to put your rubber bands across. Right now, you don't have to put it anywhere. You can just put it in the middle. Eventually, they'll go in these grooves. No, there are grooves here. Eventually, you'll put them there, and they'll help. They'll help with adjusting your microscope height. Just put them in the middle for now. And yeah, right now it's fine for them to just sit on top of this one. And same thing, you put the wing nuts and washers first, and then wing nuts. And if you find that your wing nut gets stuck in the middle, I recommend taking it out and redoing it. And because if it gets too stuck, it might like break some of the ridges. So it has not happened to me. This is pretty smooth so far. But yeah, just know that it should be pretty smooth, and you don't have to like force it through. And I'm stopping not all the way, and you'll see why.
Vanya, about how much space are you leaving between the top of the stage and those wing nuts you just put on?
Yeah, it's help.
Oh yeah,
Much
yeah.
space. Yeah. So I think approximately like your camera will essentially be like 1 millimeter, 1 centimeter, not millimeter, 1 centimeter away from your specimen, and then so the top of the stage could be around like 2 centimeters away because there's a little bit of space between the camera and the base here, and just this much space. So yeah, 1 to 2, around 2 centimeters on top, and then it's fine that it's like obviously not going to be accurate, and the first time you do it, so you can just adjust it as you go. And yeah, and then the last part of the stage is you just put the wing nuts on top, and these last 3 wing nuts you can put them in just enough that they're not going to fall off. Don't screw them in all the way, because the magic happens with the and yeah, so what these is, so now you can put these in the grooves. The reason I don't put them in the grooves beginning is because it makes it a little bit hard to like screw in the bottom wing nuts, because essentially they're taking up the same space, the wing nut and the rubber band. And so yeah, now that your wing nut is screwed in, you can put the rubber band here and lift it up so that it hooks onto this top wing nut. This is how you like keep your microscope in the configuration that you want it to be in. Or cool. So yeah, so now it's pretty sturdy. It like stays, and it will drag the top stage up to where the wing nut is. And then if you want to adjust, you will have to adjust all three and make them somewhat parallel to make it go up and down. But right now, we'll keep it here for now. And to do the last part of the stage, all you need is a piece of, I just use printer paper. You can use a Post-it note or anything you want, just a white piece of paper, and you can put it on the bottom. And then probably best to just secure it with some tape. A little bit of tape here. And if there are questions, feel free to interrupt me as we go. So I've taped down my piece of paper, and I think we're pretty much set for stage. So the next part is converting the webcam. So this is just a regular webcam that you get with the kit. So what we need to do here is pretty simple. And basically the thing is to take this out, but we will need to open up the webcam and take out this top cover and then put it back together. That's what we're gonna do now. So two sides, pick the plus side. And then there are two, depending on what kind of webcam you get, you might have 4, but this one has 2 screws. And I find it easier if you like open it up, so this part is not in the way. Make sure you don't lose the 2 tiny screws. What you can do, so once that is unscrewed, then it comes apart. And then you can just pull this off. You don't really need to twist it. Gently pull it off. And just come off. And then, yeah, we're good. And then, so what you will want to do here now is you can focus it a little bit. So I've already somewhat pre-focused it. So just unscrew it 2 turns approximately, and then you can try to find something with small print. I think the thing I can find is like eye drops, and you can plug this into your computer and try to focus it so that your viewing distance is around 1 centimeter, like around the square parts. So I won't demonstrate the focusing part, but I think it's all you need to do is plug this into your computer and then focus it a little bit, and then eventually we can, if needed, we can further refine focus at the end. So yeah,
Vanya, are you? Oh, sorry.
Yeah, go ahead.
Can I ask a question? Are you recommending to do like a little bit of focusing while you have the camera just in your hand like that before you place it on the stage?
Yeah, it's a little bit easier. Well, it's a little bit easier to like focus now before you place it on the stage, but you can definitely like place it and then trial and error figure out if you need it to be closer or further away. Yeah, but I do recommend doing a little bit of focusing here. So unscrewing, like unscrewing it around twice, and that will get you pretty close. So I think I found the more you unscrew, the closer you can get to your specimen.
Cool.
Yeah, I've already turned it around 2 times.
Okay, thank you.
Yeah. And then now you put it back together. And I think this is actually probably you can put back your screws. I recommend putting in both screws at the same time before screwing them in, just to keep it in place. It's not going to fall off, not going to open up, so good enough for me. And then so this is where your microscope will end up looking like, with the camera side down. And then to secure it, you'll just put a couple pieces of tape to tape it down. The tape on the side of the microscope or the webcam. Face. There you go. That's pretty much it. A couple other things to note is that you will have this light and the way you activate this light. I just have like a battery pack here, and you can plug this into your laptop or any source of power with the USB. And you just tap the top and wait. All you do is tap the top and it'll light up. And then what you will need to do is kind of adjust the distance to the point where you can see your specimen clearly. And I find that it's you do need quite a bit of distance, around like at least 3 or 4 times the distance of the height of the microscope that you need to put your light at, but it's something that you will need to test out yourself. I think that's it. And all you need to do is plug in, so I can plug this in to be able to see anything. Okay, now I've switched to my microscope, and let me see if I can. It'll probably be pretty blurry, and this is where kind of the adjusting comes in. Just have a quarter. And yeah, so obviously it's pretty dark, and that is why we would like.
We can see it though.
Yeah, not the most optimal light. Let's see if we can find some text. We found a little bit of text.
That's actually pretty clear.
Yeah. Yeah. Okay, pretty good for like no adjustments.
Yeah. Can you show us how we would make adjustments if we needed to?
Oh yes, I can. Let me switch to my other camera. Too many cameras. Yeah, so the way you would make adjustments is to turn the wing nuts, either like unscrew them or screw them in. If you want the stage to be lower, then you would turn them all clockwise and make sure to turn all 3 so it's not like super weird and tilted. So, and so that's kind of just what I would do. 3, and then a little bit, and it'll be closer. Yeah, that's it.
Cool. And I can see also that your sample is on the lower stage so that the camera is above it.
Yes, that's right. So it's on top of that white piece of paper. On the
Yeah.
Yeah, and that's pretty much it. I think it's pretty simple. but yeah, super fun, and you can put a lot of things underneath. And I know Alejo will be demonstrating things that you can put underneath your camera, but some things is like you can put pollen, you can put the pond water. The light microscope might be better for pond water. You can put like mushrooms or food or plants or leaves. It's pretty open to your imagination.
Awesome. Thanks so much, Vanya.
Yeah.
That was super clear, and I'm so happy we got to see your quarter.
I have a quick question.
I am. Well, yes.
I have a quick question. I think I missed maybe just one little bit. Can you repeat, Vanya, what the, like you had the eye drops for? Was that just to help?
Oh,
like when you were.
So I just wanted something with small font or some to focus the camera a little bit. So when you undo your webcam, you'll need to twist it a little bit to focus such that you're like 1 centimeter away from your target item. So you can like kind of see it relatively clearly. Yeah.
Gotcha.
No,
Thank you. Okay, gotcha. Thank you.
You can also use something like a ruler, or like Vanya has like a coin or a dollar bill to help make sure you've got the focus well too. Awesome. Well, thank you, Vanya. Yeah. And then we are going to, here, I'll unpin your video and then pass it over to Alejo. Oops. To show us some applications for a dissecting style microscope like we just built.
Okay. Hi again. I want to share my screen. You know, after all, I want to talk about a little about view indicators. I made a step-by-step activity in which I propose to study the value indicators to see how polluted or not are a river, stream, or creek. There is a lot of aquatic invertebrates like insects, snails, leeches, and I want to show you how they look when they are alive and in the water and swimming. And I want to share the screen. The screen, not the scream. Okay. This is very complex. I'm sharing the, no, where is full screen? It's bigger. And well, I hope you can see how it's look. Well, there is a, how is the name in English for this? A trail, yes, a trail. And you can see here that there is some things moving. This is like a worm swimming. This is very similar as how the mosquito midges swim. Actually, they are mosquito midges swimming, but are not biting mosquito. And some of them are known like bloodworms, like this. I don't know if you can see this animal is quite red, too much red. Maybe there is a little clear. I don't know, maybe I need more light. But this big and fat worm is red. I don't know if you can see that is red, but well, this is a bloodworm. There is another, not swimming, you know. They do some caves with debris in the bottom of the rivers, and they move, and then eat the same debris. They're a little red. When you see on the microscope, on the dissecting microscope, we're going to see it more red. Mainly if we have a bottom line. If there is a lot of these animals in a river or a stream or a creek, we can be quite sure that maybe there is organic pollution, like sewage, for example. But there is another animals like the mayflies, the immature stage nymphs that I don't see right now. One, well, they swim very different. There is one. Oh, I lost the focus. I don't know. There is one. I'm gonna touch it. Is on the bottom of the image, and you want to touch it. They go away. They have six legs, and they are almost all the time on the bottom. But then sometimes they swim very, very fast, like when you touch it. They swim with the tail. They have three tails, mainly. The mayflies need a lot of oxygen in the water, and they actually are very good indicators of good quality of rivers, streams, and creeks. And also they are very good to see like a good quality indicator. If there is a lot of these animals on the river or on the and there is no bloodworms, maybe the site is in good conditions. These animals are from the Suquén River in a part that is not so polluted. Then there is bloodworms and also mayflies. There are the two kinds of bioindicators. But we are going to see it on the dissecting microscope. First, we are going to see it on preserve on alcohol, ethylic alcohol. I don't find, yes. You will need a slide. You can see it because it's a piece of glass, you know. I'm sorry. I'm gonna show you some tools. That could be useful. I mean, I have this thing that I know, like a pipette. I don't know if it's okay.
Yeah, pipette.
This is like a. This is for a one use kit for odontologists. The teeth,
like dental tools.
Dental tools.
Yes.
It's for one use, but it's useful for this because we are not getting this on the mouth of anyone. They are preserved. These animals are preserved in ethanol, you know, and you will see that not all here there is a bloodworm. I want to, and I'm going to put right here on the, to see how it looks, you know. I mean, I have to turn on the microscope, the light, to see it. I'm sorry. No, this. This is a quite different microscope model, but it's, I don't know what. I see something strange, but I don't know what it is. Sorry. Just a minute. Well, it is not on focus. Give me some seconds. Is something. Some of these bloodworms or chironomids is the family for these Leeches are the focus is for the other side. I have to. This is going to be. There are a big animals. Some of them, they have more than a centimeter. But when they are big, but when they are on the final stages of development, then you can see when you have some preserved material that they have, this is the tail. Where's that? And we have the face, the head, I'm sorry. You can see there is a tiny eye. I don't know if you can see it. And well, I have to maybe to get more precise focus, but you can see that, oh, sorry, that is the head, and they have only a pair of arms. And that is different like the Mayfly. The Mayfly has 6 legs, you know. This is because there are a very different kind of insects, immature insects, but it's easy to recognize also if they are dead because they have like a C form, and they and in the end of the tail of caudal zone, they have a little gills, like a hair, like a black hair. I don't know if you can see something that comes out. Well, there is not a bloodworm. The bloodworm is a chironomid. It has the same. I'm sorry. Yes, this is a bloodworm. But when you preserve on ethanol and 70% ethanol, they lose the color. Because of that, it's very important to see, to use the step-by-step activity of the bioindicators to see it on the field alive. And then, and that's, you don't need to kill them, you know, if you don't like to do that. Well, that is a bloodworm that preserved, but sometimes it's useful to have these animals dead and preserved, because in ethanol they maintain a lot of time in this condition, and you can see it to remember how a bloodworm was, and but there was one of the last instars. There's some that are quite small, and you can see it with your naked eye. I'm searching. When they are dead, also are quite similar to snail poop. Snails.
Did
when
you just
they
say
poop.
snail poop?
Yes.
Similar, they look similar to snail poop?
Yeah, it's because the snail.
Uh-oh.
Okay. But yes, the snails, you can see, I have a snail right here. I don't know if you can see. I have this. They poop a lot. I don't know how to say this in other way. Feces is the more elegant word. Yes, they poop a lot. I don't know why they do so much poop, but they do a lot. But there is a lot of, there is another kind of chironomids that are more green. They look so similar because it depends on the stage, what tiny are they. In the first stage, after they leave the egg, they are very small and you can see it, but this is very tiny, and you only can see in detail on the microscope. And for the, I'm gonna show you also a mayfly. I say this mayfly, and you can see the three tails that they have. I'm going to use another slide. It's good to take to have, sorry, to have another tissue paper to dry the glasses to get clean. And I'm going to collect some water to put the sample, and I'm going to show you that.
Alejo, can I ask you a quick question?
Yeah, sure.
If someone doesn't have glass slides, can they use another thing to view the macroinvertebrates on, like a small like plastic lid or something like that? Like, would something like that work?
Yes, it could be work. The only caution you must have, I think, is to not this, for example, if you are going to use something like this, you can use something like this, but the walls not be too tall that maybe you can make you can get the focus, the accurate focus if the walls are so high. But I'm going to show you how a mayfly looks. Well, they look very big. But they have 4 legs right now, 5 legs, because I took and they this animal lose one when I tried to catch it. They have, this is characteristic of mayflies. They have 3 tails, and with that they swim. With the tails.
This is incredible.
And that is the eye. And well, there is also, I get fun with these things, maybe because I'm a nerd, but there is a lot of species that are indicators of some state of water quality. Also, there are leeches. I put, when I get these animals from river yesterday, and there was a leech. You know, this animal that suck blood. Well, not all the leeches suck blood, but when you take some from the river and you take another, you have to take it away if you want to live, because the leeches eat all the other animals. They are predators. I have one. Maybe you can see. And when they are alive, the leeches have a very particular moment that is they have something to suck the surface in the caudal zone, and they do that. You know, maybe if my little friend wants to do.
Beach friend.
He's leaving the glass. He's want to run away. Oh, no. He escaped. Or she, I don't know. Or they, I don't know. Well, I lose it. They move very fast.
We are all in such suspense over here, like waiting for a leech. It escaped. Where is it? Oh my gosh.
It's very big, and when you know, he's so fast. It's so fast. I lose it again. Well, he's so fast all day. He's so fast. Well, I'm going to use
Are they kind of transparent bluish color?
Well, is there? Vania, what do you say?
Yeah, I'm just wondering, is this just the regular color of leech? Is it just kind of like transparent, almost bluish?
Yes, it's like brown color or transparent, a little transparent. It's not like the big leech. The big leech are brown, very dark. They are very transparent. And well, I want to show you. Oh, it's quite late, but I mean, when they are alive, it's more funny to see. And well, I don't know if you have any question. Oh, what's it to see anymore? Anything?
I think we're all just amazed at that you've got like this next to you in a tray. You're just like showing us these organisms.
Yes, I try to put only the dead animals in the glass, because the live animals get stressed a lot of more than a live animal. It's natural, but there's a lot of things to see with this microscope. Also, you can see some protozoans, or maybe you can see tardigrades. They have water bears. They are so quick. It's so cute because they are not so easy to find. But maybe they are on mosses. They live mainly. But well, I think that is all that I have to. That is my microscope is more is quite more. It's not so sophisticated like the Vania, but it works. Well, I'm going to stop to sharing the screen. Oh, I'm sorry. What?
That was amazing.
I'm glad to hear that.
Yeah.
Cool. Now I want to go and get some pond water.
Yeah, and mosses, and I got lots of mosses where I live, so I'm gonna find some tardigrades, some water bears.
Yes, sometimes it's crazy, but sometimes people think in rainforest in which there is a lot of biodiversity, but maybe in a little pond there is a lot of biodiversity, and it's funny to see little things moving and doing stuff.
Well, thank you, Vanya and Alejo, for showing us how to build and use a microscope for this. Yeah, this is fascinating. I love this so much. So we do have a couple minutes to ask any questions and then just like, or share reactions, because there was definitely some good reactions. Yeah, go ahead, Laurel.
Yeah, no, that was so cool. And I guess I forgot how much I love just like looking at weird stuff under a microscope. So yeah, like Vanya was saying, I want to go, out into the ditch and take a big old scoop and see what's living in there. Definitely. Yeah, thank you guys so much. That was really cool. And when you say that I can find a tardigrade on moss, just like just like normal, we have a lot of like Spanish moss where I'm from. I don't know. I'll look it up myself, but I really want to find one. And I'll let you guys know if I find a water bear for sure.
I see a water bear in a sample of water of a stream, but I know that the books say that they are on mosses, on these green primitive plants. I don't know how to say it in other way. And yes, you have to try.
Wetter environments typically, although they're like infamous for being able to withstand all sorts of harsh, extreme environments. Yeah, but yeah, we can go on a hunt for our respective hunts for water bears.
I had seen them. Yeah.
Oh, sorry. I just want to say, like, one of the cool things, like, if you do find some like lichen, you can, like, if you have like mortar or pestle to crush them up and put it under a microscope, you can see the algae and like fungi kind of separate, or you can like see it like inside the fungi. It's super cool.
Oh, that will be amazing, Vanya. If you do that, I'm sorry. If you do that, please take a record and share that. It would be very, very nice to see that.
Yeah, well, I use someone else's microscope that's like a real microscope, and it's like super cool. So I'll try it with this one, and then also try it with the real one and try to take some pictures.
Great.
Can I ask one question? And this is kind of like a setup for Vanya, because you already talked about lichens a little bit, but what are some other applications you could see using a microscope, like a dissecting microscope, or even the compound version for?
I'll take a stab. Plants, lichen, pollen. I'm looking at my succulent. You can probably take out like parts of, like, if you something that fell off your succulent, like leaves from your plant.
And I'm gonna just share like a brag basically about Vanya, but Vanya's been working a lot on looking at lichens as air quality bioindicators. So Leho just covered these macroinvertebrates as water quality bioindicators. Vanya's been looking at a complementary sort of thing, but for air quality. So the microscope could potentially be used to identify species of lichens, or kind of look closely at them to see if they're responding to different air quality environments. So like she said, just lots of things you can look at under there. Cool. Okay, well, we're at the top of the hour, and this was super fun. And I am so excited to look at more with my microscope too. So thank you again for showing us, and yeah, psyched to go out there and reconvene again when we each have like different microscope images to show what we found.
Yeah, thank you for the demo. I hope it's super interesting.
Yeah, thank you all both. That was really great.
Thank you. The building of the microscope was very, very clear. And thank you, Jeanette, and thank you, Laura, for being here.
All right. Thanks, everyone. We'll see you again soon.